Our genetic roadmap is very important. Making DNA from DNA is called replication. This process takes place in the nucleus. It occurs during interphase S phase. Taking the DNA and making a working copy called RNA takes place in the nucleus. This process is called transcription. This process of transcription ends up with three types of RNA: mRNA, rRNA, and tRNA. Taking these three types of RNA into the cytoplasm to make proteins. This process is called translation. The sequence of nucleotides in the DNA molecule gives the sequence of amino acids in the protein. Therefore, the information in DNA specifically goes to making proteins. This method of storing the information for the protein synthesis is the genetic code. The RNA molecules copy and transfer the information from the DNA into the cytoplasm where the proteins are made. Why do we make these RNA copies of DNA? Well, first of all, DNA is a very large molecule and it's stuck in the nucleus, just the way a reference book is stuck in the library. You make copies, and those copies can leave the nucleus. RNA differs from DNA in other ways as well. First of all, RNA is single-stranded, where DNA is double-stranded. RNA contains the sugar ribose instead of the sugar deoxyribose, and RNA has a different base, uracil, instead of the thymine we see in the DNA. The messenger RNA carries the message of the protein. It is synthesized in the nucleus, and its sequence is complementary to the DNA template. The messenger RNA leaves the nucleus and associates with the ribosomes in the cytoplasm, where proteins will be made. And remember, this process of making proteins is called translation. Ribosomes themselves are made up of RNA, rRNA or ribosomal RNA, and proteins. This rRNA is the second type of RNA. The messenger RNA carries the protein message as codons from the nucleus to the cytoplasm, where it joins the rRNA which makes up the ribosomes. In the cytoplasm, the third type of RNA, transfer RNA, has a triplet of nucleotides called the anticodon, which is complementary to the nucleotides of the messenger RNA codon. The ribosomes hold the messenger RNA in position, while the transfer RNA carries in the amino acids in sequence. These amino acids are present all the time in the cell. The anticodons of the tRNA match the codons of the mRNA. So here we have a picture of the mRNA with its codons. The transfer RNA is a clover leaf shape, and on each transfer RNA will be a single amino acid. This anticodon ACC of the tRNA is complementary to the codon UGG of the messenger RNA. Each amino acid corresponds to a triplet of DNA nucleotides. A triplet of nucleotides in the messenger RNA is called the codon. We can identify the amino acids specified by each codon. It's a triplet, so we have a first base, a second base, and a third base. Let's say we have the triplet CCC. The first base would be C here on the left. The second base would be C on top here, and then third base C, CCC. This codes for the amino acid proline. You can see that there are actually 4 different triplets that code for proline. So CCU, CCC, CCA, and CCG all code for proline. The ribosome will contain enzymes that will join the amino acids together. The amino acids are joined. That's the primary structure of protein synthesis. And as that protein forms, it begins to fold immediately.